Welcome all to today's highlight seminar. So before I introduce today's guest, I'd just like to start with some reminders. First, please silence all your cell phones. Second, I'd like to ask you to please, when it comes time for questions, this talk is actually being recorded, so when during the question time, I'd like to please wait for you to get the microphone so that your question is recorded properly. Then after the talk, there's going to be a reception right here in the lobby, immediately following the seminar. And also I'd like to remind you that next highlight seminar will be on Wednesday, November 9th. We're going to have Stephen Cawley. He's a nuclear fusion expert from Oxford. So with that, I'd like to introduce today's speaker. He is Rybeck Lynd. He is the Paul and Joan Cuneau Distinguished Professor of Engineering and Adjunct Professor of Biology at Dartmouth College. He is Focus Area Leader for Biomass Deconstruction and Conversion at the U.S. Department of Energy Bioenergy Science Center. He's also Executive Committee Chairman of the Global Sustainable Bioenergy Project. He's also quite an entrepreneurial guest. He is a co-founder and until recently the Chief Scientific Officer of Mascoma Corporation, and more recently the co-founder and Chief Scientific Officer of Enki. So both are biofuel companies, as you might have guessed. He is a fellow of the National Academy of Sciences, and he's also received many awards. Just to mention a few, he's the inaugural recipient of the Lamelson MIT Sustainability Prize for inventions and innovations that enhance economic opportunity and community well-being while protecting and restoring the natural environment. He's also received the Charles D. Scott Award for distinguished contributions to the field of biotechnology for fuels and chemicals, and he's a two-time recipient of the Charles A. Lindbergh Grant in recognition of efforts to promote a balance between environmental preservation and technological advancement. So with that, on behalf of our community, I'd like to thank you for coming. We're delighted to have you, and I look forward to your talk.
Thanks very much. It's great to be here. I have truly enjoyed my day and look forward to sharing some thoughts and further discussions. So I'd like to talk about my only slide that's going to talk about sort of the big picture. Land and resources. Cellulosic biomass, of course, is the main product of photosynthesis, which is the organic matter income from the biosphere. New agricultural land needed to feed the world, according to the FOA, is about, in 2050, is about 70 million hectares, which is 5% of that available. Land classified as pasture is 2 times global cropland and produces about 1% of global dietary calories. In terms of climate and environment, transport modes involving long trips and heavy vehicles, planes, ships, and trucks, are on the order of 50% of total transport energy use and are difficult to power without liquid fuels. Failure to decarbonize these modes would give emissions about twice those compatible with a 2-degree centigrade increase. Biomass provides an average of 25% of primary energy supply in primary low-carbon energy scenarios for 2050. Large greenhouse gas emissions can be achieved using biofuels, notwithstanding indirect land use change and alternative land uses. There are water quality and soil fertility benefits from perennials and agricultural landscapes. I offer these as assertions without proof or discussion. This is another seminar that is not the one I'm presenting today. In terms of human development and food security, biofuels have demonstrably fostered human development in Brazil, and poverty and food insecurity, that should say, more one problem than two, can be positively impacted by biofuels, although they can be negatively impacted as well. Having said all of that, and at least acknowledged the dimension of the big picture issues, I don't claim I've settled any of those complex issues. I'm going to talk today about production technology and underlying fundamentals, and I hope that will be of interest and use to you. I'm privileged to work with a whole bunch of people, my group at Dartmouth, the Department of Energy, the USDA, Enki Corporation, and some international sources of sponsorship as well. I'm privileged to be part of the DOE Bioenergy Science Center, also a project on co-treatment, which I'll be described, and some others. Here's a not particularly out-of-date picture of my group at Dartmouth. Okay, so I'd like to start by taking a little bit of a historical perspective and talking about the thermochemical pretreatment fungal cellulase paradigm. A mouthful, I'll admit. We'll begin the story in World War II. There was at the time a unprecedented experience with canvas, natural fibers, and how they lasted in warm climates such as the South Pacific, and the answer wasn't very well, and so did not last very well. And so the U.S. Army initiated a program at Natick led by the then young Elwyn Reese, shown here many years later, along with Mary Mendels. And they worked for several decades, but then synthetic fibers emerged. And so this effort was refocused on cellulosic biomass to food and fuel. And they asked, what microbes are the best cellulase secretors? The answer they came up with was with aerobic fungi, and this is a quote from that era. Thousands of microorganisms have the ability to grow on cellulose, but only a few grow rapidly and produce extracellular cellulases capable of converting crystalline cellulose to glucose in vitro. So it's clear what these people were looking for. The other question that was asked in forming this paradigm is what can be done before biological processing to enhance enzymatic solubilization? Mechanical milling was too energy intensive. Thermochemical treatment or pretreatment was found to be more promising. And so since about 1980, the dominant paradigm in this field has been one of thermochemical pretreatment and fungal cellulase as a way to address what I'll talk about as the recalcitrance paradigm. Now, I am talking about one branch of the two ways one can make fuels out of cellulosic biomass. You can do it entirely thermochemically. I will be talking primarily about doing it biologically. And so various things have happened since then, and I'll just fast forward the tape to that. We've got about, depending on how you count them, about 5 facilities globally that produce cellulosic ethanol as of 2016. And these are all, they have common features. One of them is every single one of them uses the thermochemical pretreatment fungal cellulase paradigm. Every one of them uses grass rather than wood as a feedstock. Yeast is used in 4 of the 5, and they are all very expensive. So you see the capital cost per annual gallon of $15 being the average there. These are publicly available sources from the internet. And so let's look at this a little bit more closely. And so not only can we talk about oil at $30 a barrel, but we can also talk about oil at $100 a barrel. What did gasoline cost? What was the market price of corn ethanol? What would have been the BTU equivalent price of corn ethanol, equivalent with gasoline? And then if you look at simply the cellulosic ethanol capital cost, which from the prior table averaged $331 million, that's $15 of capital investment per annual gallon of capacity, which translates into only the capital contributing about $3 per gallon of ethanol. And so that exceeds the market price even when oil was at $100 a barrel. It's 4-fold higher than the gasoline equivalent market price at $30 a barrel. And this is exclusive of feedstock. And in a commodity process, you'd really like to spend most of your money on the raw material rather than capital. So what makes I call these pioneer plants. I could call them first-generation cellulosic biofuel, but the problem is you get into first-generation, second-generation plants. And so we'll call them pioneer plants. Why are they so expensive? Is it the cost of feedstock? In a word, no. In terms of purchase price, cellulosic biomass, even at $100 a ton, is equivalent on a dollars per gigajoule basis to oil at about $40 a barrel. And there are cheaper sources of cellulosic biomass, for example, bagasse. Is the problem that they made the wrong product? They made ethanol? No, I don't think so. Ethanol is the least expensive biologically produced fuel molecule today, liquid fuel molecule, and in my opinion will be that for the indefinite future for various reasons. As Costas Marinas commented from Penn State, if you can't make ethanol from cellulosic biomass, you can't make anything. We could go into this in more detail, but my view is that ethanol is the logical point of entry and proving ground for new lignocellulose conversion technology. In any case, there's no other molecule that we can make more cheaply today, that's for sure. So the reason these plants are so expensive, I don't think, is the ethanol. Furthermore, there are facilities that make ethanol from relatively easy feedstocks and are competitive. So the key underlying reason, in my view, is, let me acknowledge, of course, the plants we have, the facilities we have out there are first-of-a-kind plants. They can and perhaps will learn by doing. There's no question that if we built 10 of these, they could be less expensive. I personally don't think that will be enough to get us all that far, nor do I think it's very likely that these plants will be replicated, frankly. But nevertheless, I do want to acknowledge that. But then I want to talk about what I think is the key underlying reason, which is, in a word, recalcitrance. From an evolutionary perspective, microorganisms and their enzymes have, if you will, been chasing plants, but the plants have evolved to be difficult to take apart, which is manifested in various mechanistic features of the lignocellulose matrix, which I understand in Jose's class you were talking about this morning. From a processing point of view, if you're going to process biologically, the problem is pretreatment and hydrolysis, and in particular, the cost of added cellulase. So the high cost of pretreatment and cellulase is the barrier, and a trade-off that would be familiar to anyone in this field is, if you really aggressively pretreat, if you use ionic liquids, for example, you can lower enzyme costs. In principle, you can lower them to nothing if you use acid hydrolysis, for example. But you've got very high processing costs to get the feedstock to that point. Alternatively, if you want to use high enzyme loadings, you can lower the pretreatment costs, but it's really hard to get that spot where one of them or the other is not eating your lunch. And these enzymes are very expensive, actually. In fact, in general, if you look, this is just the cost of producing protein in fermenters on $1 per kilogram or $1 per pound basis. This is glucoamylase. These are various loadings in a couple of sets of units, which may or may not be familiar to you. But this is a very low enzyme loading. I'll talk in a minute about where the literature is at and where the research field is at. But if you go over here, you get to costs that are rapidly on the order of $1 per gallon of ethanol for the catalyst. And you get to the point that the catalyst is costing you more than the raw material. And so it turns out that most of the research literature is up in this area, which is really astronomically high. And you'd really like to be down here. And even then, getting to 50 cents per gallon of ethanol for your cellulase cost is a fairly heroic thing to do. And that's, again, a catalyst comparable to the cost of feedstock is really unprecedented and unlikely for a commodity process. And it's not getting any better. This is a historical view of published estimates. I note this is a log to the base 10 scale of published estimates for the cost of cellulase enzymes, starting to get some actual industrial values here, and they tend to be in the 50 cent to $1 per gallon range. So the title mentions new questions and new answers. So here's one of the relatively new questions. What are nature's best catalysts at deconstructing cellulosic biomass? So acknowledging again the pioneering work of Rees and Mendels and their colleagues, but it was undertaken in a pre-genetic engineering era. They were explicitly looking for the best cellulase secretors, which is not the same as looking for the best systems for taking apart this stuff. There have been a few biochemically oriented enzymatic studies, generally on model feedstocks, often emphasizing initial rates, which indicate that complexed cellulases have specific activities on the order of 10-fold higher than non-complex cellulases. What do I mean? Well, here's a cartoon of a non-complex cellulase. You have the protein, the catalytic components operate independently of each other. And then for the complex cellulases, typified by Clostridium thermocellum, which I'll be talking about quite a bit, these have these multi-catalytic machines. If you think about what that looks like, so here we have an actual photo of yeast cells, not cellulase molecules, and a cotton fiber. And so if you're adding enzymes, which are not shown here, they would be too small to see, the cells have no particular relationship to the cotton fiber. This, though, is the way that nature does cellulose solubilization, using complex enzymes expressed on the surface of the cell. So what we would really like to know is, what are the comparative hydrolysis yields? That's more important than initial rate. What are the comparative hydrolysis yields on lignocellulose? This sounds like a simple question, and in some ways it is, and it could have been asked 30 years ago, but wasn't, by and large. But it is technically nuanced. You can't just do one condition and sort of call it done. It's never been easy to get funding for. There's never been a time that doing these sorts of comparative studies was sort of the easiest thing to write a proposal for. And interestingly, if you do 10 of these and you show that one's the best, you may have 9 people who resent you and 1 who's pleased with you. And so there are a whole lot of reasons this hasn't happened, but it's a rather foundational question. And so we looked at this in a paper that came out earlier this year, and we did a comparison where we looked at what happens if you just incubate the stuff, and this is without thermochemical pretreatment now. And we looked at some mesophilic microorganisms, we looked at fungal cellulase and yeast, we looked at a horse manure enrichment, which we expected to do better than pure cultures, but in general, in this and other studies have not. And then Clostridium thermocellum. We did lots of controls where we reproduced results from the literature and made sure that we were, with systems we had varying degrees of familiarity with, getting things similar to those who had more familiarity. I could talk more about this, but one of the surprises was that some of the pure cultures did as well, or perhaps a little bit better, let's call it as well, as when you let the microbiome sort of have at it. We were very surprised by that. But here's another comparative observation, which is that for this particular feedstock, the thermophilic anaerobic microorganism here did somewhat more than twice as well as fungal cellulase and yeast, which is representative of the current industry. Well, rather than just stop there, we dug into this, and we said, okay, we could add different amounts of protein. What do things look like? And so here's the percentage of solubilization of a grass feedstock without pretreatment as you add more and more protein, and you sort of hit a limit, in this case, at about 70%. We'll talk about ways to get higher than that. And to my surprise, frankly, I expected as we added more fungal cellulase that this would just keep going up. But we hit a limit at something below, or maybe at about 40%. And so again, you see the thermophilic system being superior, the bacterial system. And also, by the way, the first one we did was to have simultaneous saccharification and fermentation, so we had yeast present. But then we thought, well, hold on, maybe we'll leave the yeast out. Does that matter? Nope, about the same. And we thought, well, let's do it at 50 degrees centigrade rather than 37, about the same. And then we thought maybe there's some product of the reaction that is inhibiting it under these conditions. We'll lower the feedstock concentration, a little worse. So we gave it a lot of chances to succeed. And if you look at the solubilization by the bacterial system compared to the solubilization with the industry standard fungal cellulase, everything lies above this line. If you look at the ratio in the dark symbols, you find that the more recalcitrant the feedstock is, that is, the lower the solubilization with the standard system, the greater the relative advantage. And so it's on the order of 3-fold more effective. And, you know, we got lots and lots of data on lots and lots of feedstock. I think we've really given this a pretty thorough working. So the green is Clostridium thermocellum. The dark brown is unaffordable levels, the light brown is unaffordable levels of fungal cellulase. The dark brown is potentially affordable, although still expensive levels. Whole bunch of feedstocks. So although this is not widely appreciated yet, we think that the industry has been based on less than biology's best. My point is not that C. thermocellum is the unique best, but that there are better microbial systems at taking apart lignocellulose, better biocatalytic systems than fungal cellulase. We've done 5 different feedstocks, 10-fold range of catalyst loadings, 10-fold range of particle size, 10-fold range of substrate loadings, presence or absence of cells, incubation temperature. We have yet to find a condition where the fungal cellulase does as well, or even approaches as well as the thermophilic bacteria. Well, why is this? So it has to do with a number of factors, we think. I'm not saying we have it thoroughly figured out. Keep in mind that the primary catalytic entity is different. For the case of enzymatic solubilization, it's an enzyme substrate complex. In the case of microbial solubilization, the microorganism is a physical part of that. There's quantitative evidence for synergy that's enhanced by the presence of the microbe. The local physical chemistry is impacted by the cell surface because the reaction happens in this sandwich between the surface of the cellulosic biomass and the surface of the cell. There's also been selective pressure to optimize those surface properties. Interestingly, so these are scale models of the primary cellulase component of Trichoderma ricei, an aerobic fungus, which is used for the industry. And this gigantic thing is, to scale, a model of the C. thermocellum system. Not that long ago, people were talking about pore size as being the determinant of whether cellulase systems would be effective. And the idea was that you needed to have a small enzyme and big pores in order to get good accessibility, and good results. It's pretty obvious that's wrong, because here we have a very large enzyme system, orders of magnitude larger than this little one, outperforming it. So something else is going on. And so these are work from my colleagues at NREL, and they showed that as the solubilization proceeds, that for the fungal cellulase tends to sort of sharpen the end of the pencil. It takes these fuzzy ends and but doesn't unravel them. By contrast, this much larger molecular machine is able to physically unravel. These are actual TEM images, and here's a mutant that's lacking one of the proteins that's involved in splaying these ends. And so it's as if you've got to be a large molecule in order to sort of get the wedge in the end and get access to new stuff. So, can high solubilization yields be obtained relying entirely on the saccharolytic capability of C. thermocellum? For some feedstocks, perhaps. For paper sludge, corn fiber, which the industry is very excited about, and even corn stover, you can get pretty large in green solubilizations with the thermophilic system, much lower with the fungal system. But in general, not for most cellulosic feedstocks. For agricultural residues, for woody and senescent herbaceous energy crops, I think one could look for a lifetime or many lifetimes, and you're not going to find microorganisms that without some help can thoroughly take apart lignocellulose to the extent we'd like. So the question is, how do we give biology a hand? The conventional answer is thermochemical pretreatment. We add chemicals and/or high temperature. Well, you could do that, but I point out that, first of all, if there's one unit I've spoken to operators of the cellulosic ethanol plants that are out there. If there's one unit operation they'd like to get rid of, it's pretreatment. And it's expensive. The other thing is we have to look, once we sort of undergo this mindset change of looking to thermophiles to do our processing, thermophilic anaerobic bacteria, we have to look at what they're good at and what they're not good at. And so yeast is among the most resistant life forms to chemical inhibition. Our cellulolytic anaerobes are not particularly resistant to that. However, mechanical disruption, gram-positive microorganisms are small, tough, with a thick wall, and they are among the most resistant cellular life forms. We can also take inspiration from, let's go back. Now, let me see, this worked before. I don't understand. Where is the pointer on this thing? Well, you'll have to take my word for it. My son is a dairy farmer.
It showed up, the arrow showed up.
Oh, you don't have to take my word for it, although I can't see it on my screen. Now, let's watch what this cow's doing. So she's not eating grass, but she's chewing. And she's not chewing up and down, she's chewing sideways. And what's going on is that the grass is going back and forth between this mechanical agitation that makes new surface area available and the bioreactor, i.e. the rumen. And so this is the solution nature has come up with. And we thought, maybe we'll try to imitate that and see what happens. And the initial configuration is not the one I would recommend to do this with, but we said, look, let's run a little fermenter, let's fill it full of stainless steel beads and beat it and see what happens. Well, the first question we asked is, to what extent can the biocatalyst withstand this aggressive mechanical environment? And yeast, the sugar fermentation, is just totally stopped if you mill and proceeds in the control where you don't mill. And in the case of the bacteria, they don't really much care. Fermentation, as I'll show you, proceeds readily in the presence of milling. So we then said, okay, now let's compare fermenting twice, without ball milling before both inoculations, or ball milling between the inoculations. And interestingly, we found that the green is milling the partially fermented material between inoculations. On a log scale, we got much smaller particles, so more effective milling. We also got more effective solubilization. We call this co-treatment. So here's when you mill beforehand, you get some enhancement from milling, but here's where you mill once biology has a chance at it. So this is indicative that milling partially fermented material is different from milling unfermented material. And so if you look at switchgrass, we find that if we ferment the material without, if we ferment the switchgrass, we get about 45% solubilization, which is a lot. This is a genetically modified switchgrass, which is easier to solubilize. We get about 61. But if you co-treat, you get 92% solubilization, which is essentially, well, it's 92%. And if we if we look at gas production, it's from the fermenters, you can see it's quite well proportional to what's going on. Somewhat to our surprise, we did the same thing with poplar. So here we have two poplar lines of different recalcitrants. And we found that if we milled during the fermentation, we got 96% of that poplar solubilized. Keep in mind, we are not adding enzymes and we are not doing thermochemical pretreatment, the two things that make this process so expensive. So we've done a lot of data on this, which we will be submitting for publication within the month. Once again, here's the conventional system, simultaneous saccharification and fermentation. There's been a great deal of effort to developing less recalcitrant cellulosic feedstocks, but the impact is not that large. That's the purple here. Here we see a bunch of C. thermocellum, all of this on switchgrass, a variety of different switchgrasses. So the red here is much higher than the conventional system. Here is the best, here is the genetic variant that had the biggest difference. It makes some difference, but if you co-treat, you barely see the difference, actually. And here is the poplar results, with and without, with a more or less recalcitrant initial feedstock. You see the initial recalcitrance doesn't make much difference at the end. And then down here we've got corn stover, which we can get 93% solubilization of with co-treatment, only 21% with the fungal cellulase, and corn fiber, where we get 95% solubilization without co-treatment. So we're seeing some very high solubilization yields. The main question, which we haven't answered totally yet, has to do with the energy required for milling. But this energy is determined by two things. You have to pay energy to overcome the slurry viscosity, and you have to pay energy in order to actually break the particles. And well known to anyone who has worked with the biological solubilization of cellulosic biomass is this pronounced liquefaction phenomenon. In the first even 5%, but 5, 10, 15% of solubilization, you get radical changes in the physical properties of the material. So for example, here we look at viscosity, which declines by about 30-fold in the first 15% of solubilization. And here we look at the particle strength explicitly as a function, this is of the extent of hydrolysis. So by the time you're down to 12 or 15%, You're at a very small fraction of what you began at. And so the thought here is that if you want low milling energy, you need reactive material. If you're using fungal cellulase and yeast or fungal cellulase without pretreatment, you've got a hard time getting into this zone. But if you use these biological systems that are much better at solubilization, you can get into this region where the milling requirements aren't so high. And so rather than non-biological disruption before biological processing, which is pretreatment, perhaps we should process biologically first, like the cow, and then do the non-biological disruption or co-treatment afterwards. So here are some data for the fungal cellulase system. Here is the fraction of the material that's solubilized. Here's the milling energy. Keep in mind the heating value of the biomass is about here. And so this shows if you pre-treat with milling, you have energy requirements that are much too high. However, I've shown you that C-therm is 3 times more effective at solubilizing the biomass. Now, we're not going to triple a 50% yield, obviously, so it's not a linear world. And also that co-treatment is 2 times more effective than milling as a pre-treatment. But in addition, I've shown you 30-fold difference in the physical properties that should determine milling energy, and to top it off, these milling tests were done with laboratory scale equipment, and according to the vendors of that equipment, there's about a 10-fold lower energy requirement for milling at industrial scale. And hence, there's an awful lot of reason to think that we can head towards the bottom edge of this corner, although we have not proven that yet by any means. So, let's go back to this idea of the paradigm. This is the slide I showed you before. If we want different answers, we need to ask different questions. So whereas it was previously asked what microbes are the best cellulase secretors, and the answer was aerobic fungi, we have asked what are nature's best biocatalysts at deconstructing cellulosic biomass, and we get a different answer. Whereas it was previously asked what can be done before biological processing to enhance enzymatic solubilization, we asked what if we ferment first and then mill partially fermented solids? And again, we get different answers. And let me acknowledge that in addition to new answers and things that look good, when you go away from the conventional, you also have new challenges. And so I like to think about lignocellulose processing biologically this way. There's a front-end problem of going from this recalcitrant stuff to something fermentable. This is extracellular, and I've made an argument that thermophilic processing with co-treatment is a different way to do things that has many attractive features. Then there is the back-end problem, or set of challenges or opportunities, if you like. So it's intracellular, and let's face it, yeast are really good ethanol producers, thermophilic bacteria are not, and so you've got a whole host of issues, every one of which we have spent a great deal of time looking at, and I could easily populate an entire seminar going through this list, and have done so many times, and I'll give you a flavor of it without all the details. But to be clear, to exploit the distinctively advantageous front-end related features of thermophiles, we need to address these back-end related challenges, which involve using and developing non-model microorganisms that do not have a track record of industrial application for which molecular techniques were, until recently, non-existent. It's pretty different to talk about doing genetic engineering in E. coli or yeast versus these bugs. 10 years ago, there were no genetic systems at all for these microorganisms. And so here is an early success story. We're dealing with branched metabolic pathways like this, and we said, okay, let's knock out the genes associated with the products we don't want. We developed this hemicellulose fermenting, but not cellulose fermenting microbe in a PNAS paper in 2007. It produces essentially theoretical ethanol yield, in this case from the pentose sugar xylose, and it produces economically recoverable ethanol concentrations. And if we think about where we'd like to be up here at high ethanol concentrations. At a certain point, you can't make more ethanol than this because of solids loading constraints and material handling. So you'll never make ethanol at the same concentration as corn ethanol. And T. saccharolyticum, the hemicellulose fermenter, is where we need it to be up in this corner. C. thermocellum, the cellulolytic organism that I've been talking to you about, is not as far, but progressing and will continue to progress, I'm sure. Interestingly, if you're talking about a bolt-on plant where you're putting your facility at an existing plant, I think the window gets bigger, especially to the left, and C. thermocellum may be starting to nibble there. So we're looking at the pathway of this hemicellulose fermenter that works so well. It's not a conventional ethanol-forming pathway. It involves this enzyme that takes reduced ferredoxin and NADH and makes 2 moles of NADPH, which we believe are the electron donors, which are used to form the product. One of the problems we have, that one has, which I think we discussed a little bit in at least some of our discussions today, is that a lot of these microorganisms have multiple genes which are annotated to do the same thing. And so in this case, for example, we have 6 alcohol dehydrogenases. Well, we've gone through knocking out each one at a time to determine what they do. And interestingly, we find that the effect of given knockouts is different in the wild type and in the strain that has been engineered to do what we want. We are, and again, I'm just trying to give you a flavor of this rather than going through every detail. But we are transferring that pathway from the high-functioning organism to the lower-functioning microorganism. And so here, as you express various of the genes from the pathway, we find the single gene that makes the most difference, which is this ADHA, and then we're doing them in various combinations. So we're trying to import this pathway from one microorganism to another. And interestingly, we find that the resistance, the reversibility or irreversibility improves as we start adding more and more enzymes. Even though the yield's not getting much better as you add more enzymes, the ability to force the product out in a high external concentration is improving. So we think a lot about electrons and reducing equivalents in anaerobic metabolism. You've got NADH, you've got ferredoxin, which is formed by well-described reactions. This enters a pool of reduced electron carriers that are interconverted. And then there's a whole bunch of different reactions which, since you're an anaerobe, you have to conserve your available electrons. You can't dump them on oxygen to make water. And we're not doing respiration, anaerobic respiration either. Interestingly, one of the main co-products we make is amino acids. And that's because they are themselves electron-accepting reactions. So we're trying to sort that out. And I'm happy to discuss biotechnology details to those for whom that's interesting. But let me zoom out now. So I've talked to you about starting with nature's best and giving biology a hand. And this leads one to this idea of co-treatment enhanced thermophilic fermentation, no thermochemical pretreatment, no added enzymes. And I'll be showing you a bit of economic analysis, I think potentially transformative, although still some uncertainties to be resolved. But this also gets you into the game of metabolic engineering non-model or non-standard microbes. And again, totally separate from biofuels for a moment, some industrially desired microbial phenotypes are too hard to move. And I believe lignocellulose solubilization is likely one of those. That has rather profound implications. It means instead of doing biotechnology on a few workhorse microorganisms where you import diversity of nature by bringing genes from elsewhere into those bugs, it means you actually have to do biotechnology on these non-model microbes, which it's an interesting intellectual frontier. It's a challenge. You do things in a different order, different challenges, different opportunities. So in a way, if you have a process now where pretreatment and fungal cellulase addition are the reasons it's so expensive, and you have an alternative that doesn't involve either one of those, you might expect at an intuitive level to have the potential for much better economics. And so to some extent, this is a self-fulfilling prophecy. I'm going to show you some economic comparisons. Let me also acknowledge right now, what I'm going to do is compare to a current or most recent published NREL study of cellulosic ethanol. That study was intended to embody what can be done today. I'm comparing it to things that we can't do today, and we may be able to do in the future. So there again, you would expect, and I will show you, that you get very different and very much better economics if you compare a high beam future alternative to a low beam present alternative. But nevertheless, let's go through the exercise. And so this is the NREL base case, and this is the advanced case, which has no pretreatment, no added enzymes. But we also made another change. We wanted to try to explore making a process that was not scale sensitive, because those plants average $330 million for a relatively small plant. That's a tremendously high investment. We think we need to learn at smaller scale. And so instead of co-generating electricity, we put in fuel pellet co-production and a few other changes. But strategically, we made some changes due to R&D-driven assumed progress, and we made some changes due to configuration. And so if you look at the installed capital cost, about twice as high for the base case. But interestingly, as a number of people have noted, current cellulosic ethanol plants have about half the capital, which isn't directly involved in making ethanol, but is wastewater treatment and steam provision and cogeneration. By contrast, in the alternative we're analyzing, and we're doing this analysis, by the way, cooperatively with the National Renewable Energy Laboratory, these so-called off-site battery limits are vastly smaller. That also has the advantage, by the way, that in general, these dark technologies are mature, and the various shades of blue are not mature. You want to avoid having a process that has a lot of capital for mature technology, but for the whole thing to work, you need to, you have some risky pieces. So what you'd like to do is test the risky pieces without having to pay high-priced capital for low-risk technology. You can look at OpEx, and again, the advanced is somewhat lower. It's popular in the field to look at minimum ethanol selling price, but I much prefer, maybe because of my experience in the private sector, looking at some measure of the actual merit of the investment, which is quite different from looking at the minimum fuel price. And so we assume a fuel price here, namely $2 per gallon, which is either low or high, depending on how you look at it. And we look at scale. Here's the base case, the payback time in years, and here is the advanced case. And there's two things to note. One is the advanced case is on the order of 5x lower, but the other thing which is really striking is how flat this is as a function of scale. And so that's exciting. Let me acknowledge some of the uncertainties and factors that we have yet to address. We are not sure what the energy requirements are going to be for milling, so that could change this picture. There's further culture development required, especially for robustness at high solids. Material and nutrient inputs, this is CBP, is consolidated bioprocessing. That question hasn't really been dealt with, although it's been dealt with many times for other technologies. But not all of the things we've neglected are things that are likely to get worse. We've neglected some big things that I think would make it be better than this. One of them is, you can, in addition to reducing your absolute amount of investment, and I used to think that a big investment and a small investment with the same compounded return were roughly equivalent, and that's kind of what you learn in textbooks. In the real world, if you've got something new and you can test it with a small investment, that's vastly better than the same ROI on something that's gigantic, incomparably better. It's a lot easier to get $25 million than $250 million, to say the least. And so, but not only that, in terms of the integration with the agricultural enterprise, being able to do things small is a gigantic advantage. Furthermore, these are for standalone plants, and the first applications will be bolt-on plants to existing facilities. So we're pretty optimistic that this can offer very large advantages, and I don't know that it will work. But what I do know is it has entirely different ways to succeed and fail than the current paradigm. And if it's really clear to me, we have to look outside the current paradigm in order to find things that are going to be radically better. And so, interestingly, this is the energy output and input for the two processes. Because we're not co-generating electricity, we actually have to pay for natural gas and purchased electricity. And on the one hand, those could well be fossil. I suppose they could be renewable, but the way people usually do life cycle accounting, you take the world as it is for your inputs. So on the one hand, you could say we've sort of sacrificed our, I don't know, we got a black eye here because we have some potentially non-renewable carbon. On the other hand, I submit that you'd like to look at the balance between the carbon you displace with the products and the carbon going in. So in the base case, you make a lot of ethanol and a little bit of electricity. In the advanced case, you make a little less ethanol, you have some energy inputs, but you get this great big energy output as pellets, and indeed the overall process is approaching 90% thermodynamic efficient. So we worked with Michael Wang at Argonne National Lab to compare these based on the GREET model, and what we find is you get some inputs for both of them, marked in red, and then you've got displaced gasoline, which is comparable, and then you've got displaced electricity, which is much smaller for the advanced case. But you've got if you look at the total, when you count the net mitigation, subtracting the energy inputs and getting credit for the fuel pellets, I think I got them backwards. Over here, there's the fuel pellets, and over here is electricity. And you see that on a per ton basis, you get almost 50% more greenhouse gas emission reduction, even though you've got some natural gas and electricity inputs to the process. So not only on an economic point of view, but a life cycle point of view, again, the potential is there, and I already gave you my caveats. So let me close with this. Was there something I needed to say? No, I said that. Let me close with this slide in summary. A sustainable world, in my view, I did not spend a lot of time on this. I don't claim I presented the arguments to you, but it is my assessment nevertheless. A sustainable world is easier to achieve with cellulosic biofuels than without them, but beneficial outcomes require intent, as indeed with other aspects of the sustainability revolution. Current technology for biological production of cellulosic biofuels is still too costly because of the recalcitrance of cellulosic biomass manifested in the cost of pretreatment and added enzymes. Cellulosic ethanol is the best proving ground for new technologies aimed at overcoming the recalcitrance barrier. I believe that success has been declared prematurely on cellulosic ethanol. The DOE stopped working on it in 2011. Many funding agencies explicitly say they don't fund this anymore. I think that's nuts. Efforts to reduce the costs should include looking outside the thermochemical pretreatment and fungal cellulase paradigm, and I've tried to show you an example of that, which is this business of co-treatment enhanced fermentation, thermophilic fermentation. And in particular, I think we have overwhelming evidence, conclusive evidence, that thermophilic anaerobic bacteria are substantially more effective at plant cell wall deconstruction than those biocatalysts that the industry is based on today. Milling during fermentation gives very, very high solubilization yields. That's a fact. There's evidence that milling energy requirements may be low in light of considerations I showed you, but that has yet to be shown conclusively. This approach has potential for much lower payback times and implementation at much smaller scales. And the most promising development paths involve biotechnology applied to non-model microbes, which brings a lot of intellectual issues and some interesting science with potentially very high impact. One thing that's not up here is just a thought I'll leave you with before we go into Q&A. My sense in biologically derived cellulosic biofuels is that there's been a decision tree with many branches that has led to the technologies that have been implemented. And by the time you get down to the twigs in that decision tree, you've made an awful lot of choices. And I believe for this field to move forward that we need to do something which is not particularly easy. Namely, given that we have investors and sponsors who are invested in the success of the particular branches that have been emphasized, I think we need to go back upstream. I think we need to, if we really want different answers, we need to go back upstream and ask different questions and make some different choices. I don't think we're going to make this technology cheap at the end of the decision tree that we have first commercialized. So that's my overall view. It's speculative. So I'm done. Thank you. I hope you found that interesting, and I look forward to your comments.
So any questions?
Lynd Anlinger Center. Thank you, Lee, for a wonderful talk. So I had two questions, I guess. The first one was with respect to this fuel pellets. And have you accounted for burning of the fuel pellets in your greenhouse gas emission calculations?
Yeah, we assume that they're shipped to Europe and burned in power plants, which is what happens to most of them. And we've taken the discount for the transportation and all the rest of it. All of
the
that's
actual burning part too? Because then wouldn't it just be neutral? Wouldn't it be zero?
well, you burn them, but that carbon was fixed from the atmosphere to start with, so you're just returning it. There are issues, which if I'd given a different seminar, I could have talked about in great detail, with the temporal displacement of when you fix it and when you burn it and what else you could have done. But in this case, if, for example, those come from grass, which is those complexities apply to things other than what I talked to you about. If you fix a grass in one annual season, and you make a pellet out of it, you remove CO2 from the atmosphere, you make a pellet, you ship it somewhere, and you burn it to generate electricity, you haven't returned more CO2 than you removed in that year.
Right, so it should be zero. Why was it positive or negative in yours?
We are looking at the amount of displaced greenhouse gas emissions, okay? So if you wanted to talk about net, it would be zero. If your base case is the status quo, then you are saving, that's all.
So I have two.
Yeah, sure.
So the first one is, can you say anything about the separation costs? Because you didn't mention, at least I didn't see that you were mentioning it as a very big contribution to the overall cost, and that is usually used as an argument for why you might be looking at higher chain alcohols instead of ethanol.
Yeah, I think it's a mistaken argument. I think that I actually, as I've described to some of you today, I began life as a biologist, became dissatisfied with at least the environments I was in as a path, I didn't see a way to go forward as a life scientist. I'm not saying that there are no paths. From the choices I was looking at, I switched to engineering in order to get a more informed perspective on the problems that were important to solve and the problems that were not. And I was told distillation was the problem. I spent 2 years on it. I got 3 patents. Was very successful, published several papers, was expected to continue for my PhD, and I said, no, I don't think that's the problem. I'm going to go do something else. So I think distillation in general is overrated as a constraint. It's actually a rather elegant separation process. Those of you interested in thermodynamics, somewhat wonky argument, but if you look at it from an exergy point of view, you're putting heat in at a higher temperature, you're taking it out at a lower temperature, and the actual, if you look at the difference in terms of the work you've lost, it's very, very small. But to give you a less wonky answer, I think one of the advantages of ethanol over these higher molecular weight fuels is that it's easier to recover. And that is because ethanol looks a lot less like a biological membrane than butanol or farnesene or any of this other stuff, and therefore you can make it in much higher concentrations. It will always be the case, due to very fundamental reasons, that microorganisms can tolerate more ethanol than they can tolerate things that look like a membrane. So I think the advantage goes to ethanol when it comes to separation. And I don't think separation is that big a deal. I think it's been vastly overblown. If you look at the allocated cost from Envel and anybody else, it's not a big deal.
And then just a second completely different question. Is, I think you alluded to this, but what's your opinion on moving cellulosomes to model organisms? Because there's been work on that as well. Right?
Yeah, my opinion, which I mentioned briefly, is that there's some phenotypes that are too hard to move. And I think cellulose fermentation is one of them. The cellulosome has about 100 distinct proteins which are expressed in a coordinated fashion. And it goes beyond that because cellulolytic microorganisms don't just export cellulases. They express them on the cell surface. And as I also mentioned briefly, the cell surface has evolved to favor not only the catalytic reaction, but the capture of the solubilization products. What the bug's return on investment depends on. I know personally most of the world's experts that are working and have worked on heterologous expression of cellulases in general, be they cellulosomes or not, in non-cellulolytic microorganisms. And I started a company that invested over $50 million in that. And it is my assessment that none of us in this room are going to live long enough to see that approach work. That's also speculation, but I think when you have a really complex phenotype, you need to learn to do biotechnology where you find it, as opposed to trying to export or import that, depending on how you look at it. In years past, I have often been rather agnostic on that point, but I think the evidence is getting quite strong. I'm very good friends with Emil van Zyl, who spent his whole career working on this in South Africa. He's 59. He told me recently, I don't believe that approach is going to work. I'm either going to retire or find something I believe in.
Thank you for that nice talk. Your last comment about the tree and going down the wrong branches and so on, it sounded like you were saying it's time to go back to the drawing board. And I'm wondering, so it's taken 20 or 30 years to get to where we are with the old tree. And I'm wondering if it's going to take that much longer, do you think, to...
That's a good question. I think at current investment rates it could. Cellulosic biofuels have a lot of attention, getting a hell of a lot less, not had a lot of expectation. They're getting a lot less of both now. I think progress more rapid than that is quite possible, although that's speculation. And I know it's daunting to go back to earlier points in the decision tree, but that's reality. It's a good idea to accept it rather than to keep hoping that unproductive branches are going to bear fruit. But the concern you raise is a big one, and I think the world's current investment, qualitatively and quantitatively in this technology, is very misguided in light of the probability that we're going to need it in order to get to a low-carbon future. And you don't just need the technology, in a first pilot plant, obviously, proven the year before you want it. You've got long rollout and all the rest of these lag times have nothing to do with technology development at all. But I don't think that the current generation of technology is going to inspire replication. I don't think it particularly would have at $100 a barrel oil. So I realize the case for doing something different, in my opinion, has gotten stronger because of low oil prices, but I don't think that's the fundamental driver. I think the fundamental driver is those alternatives are too expensive.
I understand the biological route, but there's another route for handling biomass, which is reformation, high temperature reformation. Did you ever think to yourselves how, at the end of the road, these might compare in terms of cost?
Yeah, Eric and I were involved in a detailed project where we tried to turn on the so-called high beams and look towards the future and think about where the endpoints of those technologies might be. There's enough uncertainty in that I would say that my general approach is that we should not wring our hands in the renewable energy field about which things deserve investment. Research is relatively cheap, and I've heard reasons that many, if not all, arrows ought to be thrown out of the quiver. And if you did that, listen to all of those, we'd have an empty quiver. So I think we should pursue them both. I don't know which will be, I have my speculation. If the very small scale dependence I showed here could be realized, that would be a tremendous advantage compared to thermochemical processes, which in general require bigness. I will say this, though, people tend to look at them as competing technologies, and I think in some fundamental ways they are potentially complementary. One of those ways is I don't think we're going to see biological processing of lignin in absence of oxygen any significant. That is at least a third of the energy and often 40% of the energy in the feedstock. So one way or another, you're going to get value from that, whether it's burning or doing something more elaborate with it. Secondly, many thermochemical processes are exothermic, and in fact, to make their economics pay, you try to find value for the heat, and typically you have to export it, and find heat and power, and there are other ways to do it, I know. And the main energy input you need for biological processing is low-level heat. So you have the option to internally co-generate, and you can get efficiencies that we showed in the project with Eric. You can get efficiencies with combined biological and thermochemical processing that exceed what you could get with either one alone. So I think they should both go forward. If anybody tells you they know for sure which is going to be better, I would question they're right. Or I don't think we can be sure at this point. And I think ultimately they may both have complementary roles.
The last question.
Thanks very much for your talk. My name is Will McAlpin. I work for a clean tech finance firm, so I'm most interested in some of the economics. And it seems to me that a lot of investment in biofuels is largely driven by macroeconomic influences, right? So price of oil is going to fluctuate, let's assume, somewhere between $30 and $100 a barrel for the time being. So what's that inflection point, to your mind, for cellulosic biofuels, at which anaerobic bacteria being utilized ultimately allows biofuels to be cost competitive? I know you mentioned at the beginning somewhere currently around $3 a gallon.
It's $3 was only the contribution of capital to the selling price of current technology. Since that exceeds the overall selling price, it's kind of discouraging.
Sure.
I mean, if you talk about today's thermophilic technology, this is one of the situations. I mean, if you think about my best guess, the thermophilic option is nowhere near as developed and is more expensive than the status quo today. The thought is that as things progress, it may end up being lower than the status quo. So that's one kind of crossover point, although not the one I think you asked about. Right. I mean, gosh, in today's technology, I'm sure it would be many $100s a barrel. It's just plain it's not ready for showtime. It goes back to Eric's point, that are we really willing to back up, and are we really willing not to back up? I think that's the direct answer to your question. My guess is, if you look at the conventional approach, and you really wanted to have, NREL uses debt equity financing with the equity portion at the 10% compounded return on investment, which I doubt many of your people would have been the least bit interested in. So I think they very much undervalue capital, and they probably deal with a lower base cost number to begin with. You put those things together. But even assuming all their assumptions are right, depends a lot on the scale of the plant and all sorts of things, you're still over $100 a barrel oil. And as you well know, even if oil goes back to $100 a barrel in a few years, the memory of $30 a barrel oil is not going to go away. And I think there was an assumption that there might be a floor of something like 75. You read things like that. Well, it's going to be a long time before someone believes that again. And so I think the productive approach, I think if we wait for the price of oil to be high enough that these new things are going to kick in, we may wait a very, very, very long time. So I think there are really two answers. One is to develop really radically better technology based on new paradigms, and the other one is a price on carbon. And if you could get both going, you'd get there faster.
We'll be at least 1 for 2.
Let's thank Lee again.